Hello everyone, and welcome. I'm excited to have you here today. We're about to explore the fascinating world of the human circulatory system. The title of our presentation is Anatomy of the Human Circulatory System. We'll be looking at how blood flows through the body, the main organs involved, and how everything works together to keep us alive and healthy. Let's get started. Let's take a closer look at the main features of the human circulatory system. Blood plays a crucial role in transporting a variety of substances throughout the body. It carries nutrients that our cells need to function, wastes that need to be removed, oxygen to keep us energized, carbon dioxide to be expelled, and hormones that regulate different processes. The heart itself is actually made up of two pumps within a single organ. One pump is responsible for moving deoxygenated blood to the lungs, where it gets oxygenated. The other pump then sends this oxygen-rich blood out to all the other organs and tissues, delivering essential nutrients and oxygen. To ensure the blood reaches every part of the body, we have a vast system of blood vessels, arteries, veins, and capillaries that distribute blood efficiently. These vessels are specialized to handle different functions, whether it's carrying blood away from the heart or back towards it. In addition, we have specialized organs like the lungs, liver, intestines, and kidneys that serve as sites for exchange. These organs allow for the transfer of materials between the blood and the external environment, such as oxygen intake in the lungs, nutrient absorption in the intestines, and waste removal by the kidneys. The main function of this entire system is to move nutrients, gases, and wastes to and from cells throughout the body. It also plays a vital role in maintaining stability, regulating body temperature and pH levels so that cells can perform their functions optimally. Understanding this anatomy helps us appreciate how interconnected and efficient our circulatory system truly is, supporting overall health and homeostasis. Today, we're diving into the cardiovascular anatomy from an engineer's perspective. On this slide, we're focusing on understanding the key structures and how they work together. The heart, as you know, is central to this system and functions as a pump that drives blood through the entire body. It's important to recognize the major chambers, the atria and ventricles, and how they coordinate to ensure efficient blood flow. From an engineering point of view, we look at these structures not just as biological entities, but as complex systems with specific functions and mechanical properties. The arteries and veins are the pathways that carry blood, with arteries generally carrying oxygen-rich blood away from the heart, and veins bringing deoxygenated blood back. Understanding this layout is crucial because it helps us grasp how blood flow is regulated, how pressures are maintained, and how injuries or diseases might disrupt this balance. This perspective allows us to think about the cardiovascular system not just as biology, but as a sophisticated, engineered network that must be precisely designed and maintained. This slide sets the foundation for appreciating the intricate design and function of the cardiovascular system, which is essential for developing medical devices and interventions that work seamlessly within this complex environment. Let's take a closer look at the working of the heart valves. These valves play a crucial role in ensuring that blood flows in the right direction through the heart. Essentially, they act like one-way doors, opening to allow blood to pass through and then closing to prevent any backflow. When the heart contracts, or beats, the valves open to let blood move from the atria to the ventricles. Then, as the heart relaxes, the valves close tightly to stop blood from flowing backward. This precise opening and closing mechanism is vital for maintaining an efficient and unidirectional blood flow. Understanding how these valves work helps us appreciate the heart's intricate design and its ability to keep blood circulating smoothly. This mechanism is fundamental to understanding the overall function of the heart, which we've been exploring in this presentation. Let's talk about how the heart's rhythm works. The rhythmic contraction and relaxation of the heart muscle are called systole and diastole. These are essential for pumping blood effectively throughout the body. The heart's contractions, including both how fast it beats and how forcefully, are controlled by nervous impulses. These impulses are generated by specialized electrical cells in the heart wall, known as autorhythmic cells or nodes. The primary source of these impulses is the sinoatrial node, often called the pacemaker because it sets the pace for the heart's contractions. When the sinoatrial node fires, it causes the atria to contract, pushing blood into the ventricles. From there, the impulse spreads to the atrioventricular node, or AV node, which acts as a delay station. It holds the impulse for about 0.07 seconds, giving the atria enough time to fully contract and empty. After this delay, the impulse continues from the AV node through the atrioventricular bundle and then to the Purkinje fibers, which distribute the signal throughout the ventricles. This coordinated electrical activity causes the ventricles to contract, pushing blood out of the heart. Once the contraction is complete, the heart muscles relax passively, allowing the chambers to refill with blood from the systemic and pulmonary circulation. This entire process ensures a regular, rhythmic heartbeat that maintains effective circulation. Today, I want to walk you through the cardiac cycle of the left ventricle, which is a fundamental aspect of understanding how the heart functions. As you can see, this cycle involves a series of coordinated events that ensure efficient blood flow. First, during diastole, the left ventricle relaxes and fills with blood coming in from the left atrium. This phase is crucial because it prepares the ventricle for the next contraction. Then, as the ventricle contracts during systole, pressure inside it rises sharply, causing the mitral valve to close and preventing backflow into the atrium. This is the beginning of ventricular systole. Next, as the pressure exceeds that in the aorta, the aortic valve opens, allowing blood to be ejected from the ventricle into the systemic circulation. This ejection phase is essential for delivering oxygen-rich blood to the entire body. Finally, the ventricle relaxes again as systole ends. The aortic valve closes to prevent backflow, and the cycle starts anew with diastolic filling. Understanding this cycle helps us appreciate how the heart maintains consistent blood flow and supports overall cardiovascular health. It's the rhythmic coordination of these phases that keeps our tissues supplied with oxygen and nutrients. Now, let's explore how the heart's rate and strength of beating can actually be adjusted by two important control centers located in the medulla oblongata of the brain. One of these centers sends nerve impulses down what we call sympathetic, or accelerans, nerves. When activated, this system increases both the rate at which the heart beats and the strength of each contraction. This means the heart pumps more blood, effectively increasing cardiac output. This sympathetic activation usually kicks in during stressful situations, like when you're feeling fear or engaging in intense physical activity. In fact, your heartbeat can ramp up to around 180 beats per minute, and the force of each beat can increase so much that blood flow can reach as high as 25 to 30 liters per minute, quite a significant boost. On the other hand, there's the parasympathetic system, which involves the vagus nerves. These nerves run from the medulla to the heart as well, and their role is to slow down the heartbeat. The activity of this system is influenced by pressure sensors in the aorta and carotid arteries. When blood pressure is high, these sensors send impulses to the medulla, which then relays signals via the vagus nerves to the heart, causing both the heartbeat and blood pressure to decrease. So, in essence, these two auxiliary control centers work together to finely tune the heart's performance, ensuring that cardiac output adapts to the body's needs, whether during stress or relaxation. This regulation is a key part of how our body maintains stable blood flow and blood pressure, which is vital for overall health and function. As cellular respiration increases, the level of carbon dioxide in the blood also rises. This is a crucial point because it triggers receptors in the carotid arteries and the aorta. These receptors then send impulses to the medulla, which acts as a relay center. From there, the impulses travel via the sympathetic nerve to the heart, signaling it to adjust its activity accordingly. Now, on the other hand, when muscular activity ramps up, the muscle pump plays a vital role. It helps push more blood back to the right atrium of the heart. As this happens, the atrium becomes stretched or distended with blood, which activates stretch receptors in its wall. These stretch receptors also send impulses to the medulla, again relaying signals to the heart to respond. Together, these mechanisms show how increased muscular work and cellular respiration directly influence cardiac output, ensuring our body maintains proper circulation and oxygen delivery during physical activity. This interconnected system highlights the remarkable way our body adapts to different levels of activity to meet its needs. When we take a breath, air enters our respiratory system through the nasal passages, flowing into the pharynx, and then passing through the larynx. From there, it travels down the trachea, which splits into the right and left bronchi. These bronchi further branch out into smaller bronchioles, each ending in a cluster of alveoli inside the lung. It's in these tiny alveoli that the critical process of gas exchange happens. Interestingly, there are about 300 million alveoli in each lung, and together they create a surface area that's roughly the size of tennis courts, about 80 times the surface area of our skin. This enormous surface is what makes efficient gas exchange possible and is central to how our respiratory system supports life. This slide highlights the complexity and incredible efficiency of our lungs, which is essential for understanding how breathing sustains us. During inspiration or inhaling, the external intercostal muscles contract, lifting the ribs up and out to enlarge the chest cavity. At the same time, the diaphragm contracts and moves downward, further expanding the space inside the chest. This increase in volume creates a negative pressure in the alveoli, which draws air into the lungs. When we breathe out or exhale, these processes are reversed. The external intercostal muscles relax, and the ribs move back down and inward. The diaphragm relaxes and moves upward. The natural elasticity of the lungs then helps them return to their normal volume, pushing the air out. At rest, we typically breathe about 15 to 18 times a minute, exchanging roughly 500 milliliters of air with each breath. But during more vigorous expiration, such as during exercise or deep breathing, additional muscles come into play. The internal intercostal muscles draw the ribs down and inward, and the abdominal wall contracts, pushing the stomach and liver upward to help force air out more completely. Under these conditions, an average adult male can breathe in about 4 liters of air with each deep breath. That's known as the vital capacity. Even after maximum expiration, though, about 1,200 milliliters of air still remains in the lungs, which is called residual air. So, breathing involves a complex but efficient process of muscle action and lung elasticity, ensuring we get the oxygen we need and remove carbon dioxide effectively. All right, let's take a closer look at this slide. As you can see, the date here is December 23rd, 2025, which is quite significant for our timeline. This date marks a key milestone in our project, and understanding its importance helps us stay aligned with our goals. Moving forward, I want to emphasize that the strategies and insights we've discussed so far all lead up to this point. It's a critical juncture where we'll evaluate our progress and make informed decisions to ensure continued success. Remember, connecting these dots, our timeline, objectives, and strategies, is essential for keeping the overall project on track. So, keep this date in mind as a central reference point for everything we're working toward. In this slide, we see an electron micrograph of 2 alveoli along with an adjacent capillary from a laboratory mouse's lung. What's particularly striking here is the thinness of the epithelial cells, marked as EP, that line both the alveoli and the capillary. This thin barrier is crucial because it facilitates efficient gas exchange. In fact, at the closest point, the surface of a red blood cell is only about 0.7 micrometers away from the air in the alveolus, highlighting just how close these structures are. Now, let's connect this to the overall process of respiration. The pulmonary artery, which carries carbon dioxide-rich blood from the left ventricle, branches many times to form a dense network of alveolar capillaries surrounding the alveoli. This extensive capillary bed maximizes the surface area available for gas exchange. The tissue separation between the alveoli and the capillaries is extremely thin, which is vital because oxygen and carbon dioxide move between the blood and the air primarily through diffusion. This process depends on the partial pressure differences of these gases. Oxygen diffuses from the alveolar air into the blood, while carbon dioxide diffuses in the opposite direction, from blood into the alveolar air to be exhaled. Once in the blood, oxygen binds to hemoglobin. This pigment in red blood cells is the main factor that allows blood to carry oxygen efficiently. Meanwhile, carbon dioxide is transported back to the lungs for removal. After gas exchange, the oxygenated blood flows out of the alveolar capillaries, through venules, and ultimately returns to the heart via the pulmonary veins. This entire process underpins our ability to oxygenate tissues and remove metabolic waste, integral to respiratory function and overall health. Let's talk about how our body adjusts breathing based on activity levels. As you increase your activity, like during vigorous exercise, your cellular respiration ramps up significantly, by about 20 to 25 times the usual demand for oxygen. To meet this increased need, your body responds by breathing faster and deeper. Now, what's really interesting is what controls this process. It's primarily a rising level of carbon dioxide in the blood, not a falling oxygen level, that signals your body to breathe more. This carbon dioxide is detected by cells in the medulla oblongata, a part of the brainstem. When CO2 levels go up, these cells prompt your motor nerves to increase activity in the muscles involved in breathing, the intercostal muscles and the diaphragm, so you breathe more intensely. But there's more to the story, especially in special situations. For example, in high-altitude conditions or unpressurized aircraft cabins, oxygen levels can drop significantly without an immediate rise in CO2. Here, receptors in the carotid body, located in the carotid arteries, detect the oxygen deficiency and trigger an increase in breathing to compensate. Additionally, the level of carbon dioxide in your blood also affects your airways directly. The muscles in your bronchioles are very sensitive to CO2 levels. When CO2 rises, these muscles dilate or relax, which reduces airway resistance and allows air to flow more freely in and out of your lungs. So overall, your control of breathing relies on a sophisticated system that monitors both CO2 and oxygen levels, ensuring your body gets the right amount of oxygen and removes CO2 efficiently, regardless of activity or environmental conditions. Blood moves through the systemic arteries, arterioles, and capillaries because of the force or pressure created by the contraction of the ventricles. When the heart contracts, it creates a surge of blood that travels through the elastic walls of the entire arterial system. This surge can actually be felt as the pulse. Even when the heart relaxes briefly, during what we call diastole, there's still pressure in the arteries. Blood pressure is typically expressed as two numbers, like 120/80. The first number, called systolic pressure, reflects the pressure during heart contraction, and it's equivalent to the height of a mercury column, 120 millimeters. The second number, diastolic pressure, indicates the pressure when the heart is relaxed. Now, when blood leaves the capillaries and enters the venules and veins, there's much less pressure left to help move it along. To assist this process, the muscles around the veins act like pumps, contracting and squeezing the blood upward toward the heart. Valves inside the veins ensure the blood flows in one direction only, preventing backflow. And as the heart works harder, during physical activity, for example, its strength and rate of contraction increase, which in turn raises blood pressure. While blood pressure can fluctuate quite a bit throughout the day, consistently high blood pressure, especially during diastole, can be a sign or cause of various health issues. This condition is known as hypertension. So, understanding how blood pressure works in systemic circulation is crucial, because it impacts overall health and helps us diagnose potential problems early. Let's talk about the incredible network of capillaries in the human body. An adult human has roughly 60,000 miles of capillaries, which might sound like a lot, but when you think about it, they cover an area of over 800 to 1,000 square meters, more than 3 tennis courts. The total volume of all these tiny vessels is about 5 liters, roughly the same as our blood volume. Now, it's important to understand that we can't fill all these capillaries at once because the heart and major vessels need to stay filled and operational. So, the body cleverly redirects blood flow from one organ to another, depending on what's needed at the moment. For example, during vigorous exercise, your body opens up capillary beds in your skeletal muscles to supply more oxygen and nutrients, while reducing blood flow to your digestive organs. Conversely, after a heavy meal, the capillaries in the gut become more active, prioritizing digestion. Similarly, heat stress causes blood flow to inner organs to decrease, redirecting more blood to the skin to facilitate heat dissipation. Now, the regulation of this blood flow is fascinating. The walls of arterioles, which are small branches of arteries, are encased in smooth muscle. When these muscles constrict, less blood flows into the capillaries they supply. When they dilate, more blood can flow through. Think of it as a series of tiny valves that control where the blood goes. In times of danger or stress, the body dilates arterioles supplying skeletal muscles to prepare us for action, while constricting those leading to the digestive system to conserve energy. This redistribution is tightly controlled by the nervous system, specifically, the autonomic nervous system, which includes both sympathetic and parasympathetic branches. It causes these smooth muscles to either constrict or dilate depending on the body's needs. But it's not just the nervous system at play. Local control mechanisms also contribute. The presence of metabolites and other chemicals in the tissues can cause local dilation of capillaries, ensuring that blood flow matches the local metabolic activity. So overall, the control of capillary beds is a dynamic, highly regulated process that ensures the body responds efficiently to various stresses and activity levels. It's a perfect example of how our circulatory system adapts seamlessly to keep us functioning optimally. Let's take a closer look at this table showing how blood flow changes in the human body at rest versus during vigorous exercise. As you can see, the numbers are given in milliliters per minute for different organs and tissues. Now, during exercise, there's a remarkable increase in blood flow to certain areas. For example, the skeletal muscles see a huge jump, from about 1,000 ml per minute at rest to over 12,500 ml during strenuous activity. This makes sense because muscles need more oxygen and nutrients when they're working hard. Similarly, the heart's blood supply increases significantly, from 250 to 750 ml per minute, supporting the increased workload. Interestingly, the blood flow to the skin also increases, from 400 to 1,900 ml per minute. This is essential because it helps dissipate the heat generated by those working muscles, preventing overheating. On the other hand, some organs, like the kidneys and viscera, actually see a decrease in blood flow during exercise. The kidneys, for example, go from 1,200 down to 600 ml per minute, and the viscera from 1,400 to 600 ml. This redistribution allows the body to prioritize blood flow to the muscles and skin. Notably, the blood flow to the brain remains constant at around 750 ml per minute, regardless of activity level. This highlights how critical consistent blood supply to the brain is, regardless of physical activity. Overall, the total blood flow increases from about 5,600 ml at rest to 17,500 ml during exercise. This increase is driven by a faster heartbeat and a greater volume of blood pumped with each beat. So, the body efficiently redirects blood to where it's most needed during vigorous activity, ensuring optimal performance and temperature regulation. When blood enters the arterial end of a capillary, it's still under a significant pressure, about 35 torr, which is generated by the contraction of the ventricle. Because of this pressure, a substantial amount of water and some plasma proteins are pushed through the walls of the capillaries into the surrounding tissue space. This process is essential for delivering nutrients and oxygen to the cells and for removing waste products. The interstitial fluid, which bathes the cells, plays a vital role here. Substances within this fluid, such as oxygen and carbon dioxide, can diffuse freely in and out of the cells, allowing for efficient exchange of gases. This diffusion is driven by concentration gradients, ensuring that cells receive what they need and get rid of what they don't. Now, as blood moves toward the venous end of the capillary, the pressure drops significantly, down to about 15 torr. At this point, another force becomes important: osmotic pressure. While the interstitial fluid is similar in composition to blood plasma, it has fewer proteins, which means there's a slightly higher concentration of water there. This difference creates an osmotic pressure of roughly 25 torr, which actually exceeds the blood pressure at this end. As a result, this osmotic gradient causes fluid to re-enter the capillaries. So, in essence, the capillaries act like a dynamic exchange system. Water and nutrients are pushed out at one end, and waste products and excess water are drawn back in at the other. This balance is crucial for maintaining tissue health and overall fluid homeostasis.